Okay, so this is lecture 3, U22158, pharmaceutical formulation stability. Stability testing. Purpose of stability testing is to define the shelf life of the pharmaceutical product. And this will be the exact product that will be marketed and used by the patient. We've already discussed how you obtain stability information in pre-formulation of the product, and there are different approaches that are required at different stages of the development of that product, but exact product testing has to be carried out at some point. This is accelerated stability testing, the pre-formulation assessment, prediction of shelf life, temperature cycling and photostability testing are undertaken, and then finally long-term stability testing once the formulation is going to be released to the public. We've already discussed elements of pre-formulation assessment, so I'm not going to dwell on this slide. Prediction of shelf life by stress testing. Shelf life is usually of the order of several years. Therefore, we can't wait around several years to find out what the shelf life is going to be, so testing is too slow to be of use generally in product development to make a decision that the formulation is sufficiently stable. The Arrhenius equation allows prediction of reaction rates at proposed storage temperatures from data obtained at high temperatures. So we can, for instance, taking this as an example, formulate a drug as a solution, store buffered solution of drug at high temperatures for periods of minutes to days depending on the drug. So this is at elevated temperatures. Calculate reaction rates at each temperature, and then the reaction rates allow us to, because of Arrhenius's law, allows us to predict what the rates would be at room temperature, thereby leading to a predicted shelf life. Later, prototype or further formulations may be tested in a similar way to allow optimization of the formulation once you've settled on a reasonable range of components. Pitfalls of this approach is the precision of shelf life estimate may be poor, leading to wide-ranging uncertainties. The high temperatures may precipitate reactions that are not significant at normal storage temperatures, and high temperature degradation products initially formed, primary degradation products, may react further to other degradation products, and that wouldn't normally happen at room temperature, for instance, or at storage temperatures, normal storage temperatures. And because these products don't accumulate, there may be issues. So later on, at normal storage temperatures, primary degradation products can accumulate. If not foreseen, these products may interfere with chromatographic analysis of the formulation or put the toxicity of the products into question even. Kinetics of drug degradation may be different at different temperatures because of a switching mechanism that is possible. Dissolved oxygen concentration often decreases at high temperatures, and we know that oxygen is involved in the degradation of the product. Solid dosage forms at high temperatures often reduce the moisture level associated with the drug excipients, and that would compromise what might happen at normal storage temperatures. Excuse me. Semi-solid dosage forms are unsuitable for this type of stress testing due to their melting points of ingredients involved. However, Arrhenius equation does allow decisions to be made as to whether a formulation is stable enough to allow commercial production and at an early stage in the progress of the formulation. Temperature cycling, which is slightly different to, which is certainly different to accelerated testing. Storing the product at alternative low and high temperatures is often designed to subject liquid products to repeated freezing and thawing. This potentially accelerates physical deterioration of a product, leading to formulation instability. Temperature fluctuations may increase things like particle growth in suspensions, cracking of emulsions, precipitating a drug from a solution, and this allows evaluation of extreme temperature variations during distribution of a product. So this simulates what might happen in the distribution of products to their marketplace from site of production. Photostability testing. This is carried out at various stages during the process of putting a formulation together, as we've met before. And the photostability is provided by artificial daylight fluorescent lamps emitting long-wave UV light and visible light, carried out in a cabinet at controlled temperature, usually 25. Initial studies on the pure drug are to spread the material spread over a base of a shallow container directly exposed to light and after a period chemically analysed. So that's really taking into account the drug stability. Photosensitivity indicated by reduction in drug assay and formation of photodegradation products or colour change can occur and give you pre-warning as to whether a drug is a problem. Formulated product, pharmaceutical product, is then tested in exact packaging used for marketing, and redesigner packaging may be required to increase light protection, and the output from the lamps vary as they age in the cabinet, so should be frequently tested. 1.2 million lux hours used in photostability testing, so you always got to be mindful of the fact that the lamps can vary during that time, and it must be a parameter that's taken into account. You're working these lamps very hard. Long-term stability testing. Stress testing can provide useful information, however, before marketing the product, and needs to before marketing product needs to undergo long-term stability testing, which will be something that happens once the drug company decides to go ahead with the particular drug formulation. Store product under realistic worst-case conditions of temperature and humidity in controlled temperature cabinets or rooms. Samples are removed at intervals and tested. Assay of drug and other formulation components, antimicrobial preservatives, etc., and determination of drug degradation products are undertaken. Other tests may include pH measurements, microbial tests, and other physical characteristics during that long-term stability. You have to be aware when you're undertaking any of these things that the drug may well move into different climatic zones on use, and shelf life of the product depends on storage temperature and also humidity, and this varies from country to country, and a pharmaceutical manufacturer may market a product in several countries. So 4 climatic zones have been identified: worst case average indoor temperature and humidity, and these fall into a temperate climate, which is the UK and Northern Europe, Mediterranean subtropical climate, Japan, Southern Europe, USA, hot and dry climate, Australia, Botswana, Middle East are examples, and hot and humid climates, which are Brazil, Ghana, Indonesia, India and Philippines, etc. So these, and the figures are there, 21, 31, 31 and so on, the different relative humidities. So these have to be taken into account as well. Packaging may affect stability, therefore stored in exact packaging. Liquid products with closures stored inverted to allow interaction with closure to be detected, i.e. or example rubber vial closures, but not an ampule, there's no need for that because it's all glass. but we have mentioned earlier that some components interact with the rubber constituents of a packaging and maybe have to be taken into account. At least 3 batches of the formulation within the packaging, exact packaging to be used, allow for batch-to-batch differences, different pH, moisture content, etc. Development, smaller batch sizes are made but can have an effect on stability. So you want to do it to at least pilot scale, made with same production equipment, to simulate what will happen when you actually go into full production of the final product. Humidity is controlled for products affected by moisture, like solid dosage forms, and not needed for aqueous products in glass containers. Typical testing schedule: testing for a pharmaceutical product to be marketed in climatic zones 1 or 2, time at months at different storage temperatures, and different relative humidities. And you can see that this accelerated degradation condition distinguished from stress testing here. So this is quite a different situation. And that's it, really. Off of the testing schedule, you evaluate results. All results should remain in specification for the product: degradation, appearance, taste, smell, etc. All those things that we discussed previously that may be exacerbated by chemical decomposition. The lower acceptable limit for drug content of a product is set at 90% of the labelled content, not original concentration, because this will vary due to manufacturing variation or presence of overage, which we mentioned previously. So here we have a typical drug profile over the lifetime of a drug and its percentage potency changing. So this is drug content of a hypothetical tablet product. The regression line is shown as the solid line with the scatter points, and then there are confidence bounds outside, and what you're hoping is that points don't go outside of those regions, and therefore you've got a good solid formulation. If zero order kinetics not followed, then appropriate transformation of data is required before linear regression is performed. So data, if it's not first order, if it's zero order, then you have to if zero order kinetics is not followed, then appropriate transformation of data is required before linear regression is performed. First order kinetics requires logarithmic transformation. Order of degradation reaction determined by stress conditions at higher temperatures is performed earlier. Long-term stability testing will provide insufficient degradation to allow accurate determination of reaction order, so that has to be dealt with earlier. Minimum of 3 batches tested, as previously mentioned. Shelf life calculated for each batch separately. Shortest calculated value is selected as product shelf life. Increasing number of data points can bring confidence bound closer to the regression line and improve estimate of shelf life if these batches are statistically valid. Pooling data from batches when calculating 95% confident bound can give a longer shelf life. And only allowed if slopes and intercepts of the regression lines of each batch are shown to be statistically equivalent by analysis of covariance, which is a deeper level statistical consideration. Extrapolation of data beyond period for which data is available is not advisable. Possibility of degradation rate changing outside those limits has to be taken into account. So, summary. Pharmaceutical products can degrade due to chemical, physical and microbial causes. Chemical degradation is influenced by presence of water, oxygen, light and incompatible ingredients. And deterioration can be reduced, but careful formulation of product is necessary to ensure adequate stability. Packaging plays an important role in the prevention of degradation. Storage conditions of the product needs to be appropriate to where it's going to be sold. Adequate stability demonstrated during development before shelf life is assigned. Stress testing valuable in predicting likely stability, and long-term stability is needed before marketing.